time, and some of you already know some of the stuff I'm talking about, some of you don't know me. So I'm going to go over some stuff really fast. So really the main thing here is to kind of talk about what's new, what's in the area that might help your child that wasn't around a year ago or 5 years ago or something like that. I'm going to talk about, one of the things I'm going to talk about is DNA testing, because that's the thing that's been the biggest change in the last 5 years. It's really completely revolutionized the field, is how do you go from a sequence to the patient. So I'm going to just do this by, instead of just going over a whole bunch of genetics and physiology and biochemistry and everything, nobody wants to hear that. I'm going to talk about real individuals and how these things made a difference. This slide was put together for the MitoAction talk that I gave in Pasadena, California, just in November. So it's pretty much up to date. I was very happy with Ryan because he was my last patient with cyclic vomiting that was still vomiting. And finally got him under control. It took a lot of genetic testing to figure out what was the problem with Ryan. But anyway, he was one of those kids that had cyclic vomiting that was extremely regular. He would have episodes every, what was it, 90 days. Sometimes 88 to 92 days. Mom had a calendar going back many years. It never varied. It was almost exactly 90 days he would go into an episode, and nothing would make any difference whatsoever. He also had some other issues. Almost nobody has just one problem. He's had some cognitive delays, chronic fatigue, frequent urination, constipation, obstructive sleep apnea, etc. And then he was also having some aggressive, explosive behaviors, which is a very significant problem in this population. And he failed treatment with everything. I'm okay. And what we did is that we found we sequenced all the genes, and we found a variant of interest in the sodium 4A channel. SCN stands for sodium, CN, S for sodium, CN for channel 4A. So it's sodium 4A channel. I'll get to a little bit later about what that is, but it's basically, it was a variant that's present in 1.2% of the population, so it's by no means the only cause of his disease. But I think it's the main reason why he had the vomiting episodes in a very periodic manner. And he also had that would be a channelopathy. He also had another variant in a Mars gene that it makes proteins. And then based upon that, I had several other patients that had variants in sodium 4A and some of the other related sodium and calcium channels. So I put them on a drug, acetazolamide, also known as Diamox, which is, until I started using it a few years ago, nobody had ever thought about using it for cyclic vomiting. It's a water pill, it's a diuretic. But it does act on the sodium 4A channel. The episode stopped. It's now been 1.5 years, and he hasn't had one at all. So we can pretty much say that he's in remission. So that's one of the situations in which we had done genetic testing and didn't show very much, but then we did more modern genetic testing. We tested everything. And then the main thing to learn from that is that it's not just so you look at the genes and you say, okay, this is something that's clearly a mutation, this has never been seen before, this we can prove causes the disease. Most of the cases I'm going to talk about are polymorphisms. They're common or uncommon genetic findings, which are probably not the full cause of disease, but may or may not be contributing to disease. I'm going to go on a limb here, but I would say with pretty high certainty that most of you in this room, that your children or yourself, if you're an adult that's affected, do not have a single disorder, a single diagnosis. There's not one change in the DNA that causes what's going on. But there are several different changes in the DNA inherited from both parents that together, that combination contributes towards the disease or confers genetic risk. And then there's environmental components as well. And it's not always looking at the DNA to find the cause of disease, but it's also looking at the DNA to find contributing factors that can be treated. So this is about the things that I do. I'm sort of schizophrenic right now. I wear many different hats, and I'm wearing about several different hats in any given situation. My main thing is a day job as I work as a clinician. I'm mostly a physician seeing patients. I have a private practice in Pasadena, and I also work for CNNH, which is in New Jersey in the Philadelphia suburbs. So I do a lot of stuff by telemedicine. I also have an Arizona license, and I do telemedicine to Arizona as well as to Florida and some other states. It became just too difficult to drag everyone out there to the Los Angeles area. A lot of kids couldn't travel and couldn't be seen very often, and also there was the issue of that it's just an economic burden on families to bring the whole family out there with the flights and everything. So I started getting to try to do things by telemedicine. I think that that's been working really well. Also, I'm going to talk about neural needs a little bit at the end. My formula over there, spectrum needs, if anyone wants to do a taste test, it could also, there's a few of them to buy here. I can give you a special rate that we give at conferences. I worked as a medical director for Cortigen, which is now no longer sequencing. I've worked for 3 other genetic sequencing companies at different times. I do that as a medical director, so I'm very much aware of what the laboratories do in that. At this time right now, I don't work for any labs. I negotiate prices with all of the labs. So the labs that produce extremely good product, and they do that quickly, and I can negotiate them down for price. So I can get much lower rates than the regular doctors can to do sequencing. I do expert witnessing in legal cases, unfortunately, a lot of Munchausen by proxy, fictitious disorder, medical child abuse, whatever you want. And then I still do some research, still write some papers, but that's mostly in the past. For 20 years, I was at USC as a professor at a children's hospital. Still working with Dr. Frye, though. We're still doing some research together. So, Peyton wants her name being used in her photo, and she said she's actually local here. And she's one of the ones that I talked about really quickly. She got sick with vomiting syndrome, chronic pain throughout her body, chronic fatigue syndrome, GI problems, a lot of the functional issues. But the main thing she had was chronic fatigue. The chronic fatigue was so severe that she would, just to get up to have breakfast, that would wipe her out completely. She was a good student on a laptop and was able to keep up with classes, but was pretty much bedridden and unable to do anything for years. In her, we found that there was a polymorphism that's present in 1% of the population in the TRAP1 gene, which is the mitochondrial chaperone. So it's in the mitochondria. What it does is it protects other proteins in the mitochondria against situations of high oxidative stress. So we do find it in patients that have a lot of mitochondrial DNA problems, and then they have the TRAP1 on top of that because they can't. It's, again, polygenic, many genes together. If you have a mitochondrial DNA abnormality that's making a lot of free radicals, this is a situation in which you cannot protect against free radicals, and so the free radicals cause more damage. I had a lot of patients with this polymorphism, or this genetic change, which is seen in 1% of the population, and they all had pretty much exactly the same symptoms. They all had chronic pain, chronic fatigue, and GI symptoms. So based upon that, I worked with a collaboration with people at Georgia Tech, and what they did is take a three-dimensional view of the TRAP1 protein, and then did it on that was based on X-ray crystallography. So we actually had a three-dimensional picture on a computer as to what it looked like. And then the computer estimated what it would look like with the mutation in it, and how it would change it. Then the computer put in every medication that's ever been made into the binding site of TRAP1 where the mutations are, and to see if we can find something which destroys the TRAP1 binding site in the mutant molecule, but leaves the wild type of the normal molecule alone. And we found Kytril, which is a medication which is used for chemotherapy-induced vomiting, that I used before in sick with vomiting syndrome, and that it fit perfectly into the mutant binding site for ATP, but it did not affect the wild type or the normal binding site. We gave her Kytril, and her symptoms went away almost dramatically. Fatigue's still there, but less than half what it was before. Chronic pain, constipation pretty much went away. To show that what you can do when you put everything together. I had a lot of patients with TRAP1. And if they had that mutation, they tended to get better on Kytril. If they had other mutations, we would still give them the antioxidants at high dose. But I also noted that a lot of them were girls and their brothers had autism or ADHD. And there's a question of, it's not that TRAP1 causes autism, but is it contributing towards it? And so we tried treating a couple kids that have the 253 polymorphism, the TRAP1 gene, with autism. And actually it made a big difference. In Carter here, he became much more talkative, more focused, more clever, less aggressive. The main thing is his hyperactivity went away. He would be the kid that would run around and scream, and they couldn't take him anywhere. There's no one that can babysit him. Now he can actually, they can go to a restaurant and people sit down and eat. So it makes a big difference. Kytril is an expensive drug. Insurance, this is way off label. Insurance did not want to pay for this indication. They kept denying it, and also pharmacies started running out of Kytril because it was a nationwide shortage. So he was taken off of it for 4 times, not on purpose, but because the drug was unavailable. And he regressed each time, and then he went back, and when he went back on, he got better again. So it's not coincidental. Again, this is in mitochondrial disease, and also in pediatrics in general, almost everything is off-label. That almost everything I'm going to talk about, without exception, are not FDA approved for these things. So this is my requirement if there's anyone who's an FDA plant in this room. Never know. Okay. We go through some of these really fast. I don't have a lot of time. Carl is a young adult. He was having episodes of abdominal migraine that would cause extremely severe abdominal pain. I've seen him in 2 of the episodes. He would be literally like jumping off the table, like someone with an appendicitis or a kidney stone, screaming and begging for relief of the pain. Between episodes, he's a completely normal, guy, and he, in a major metropolitan area in the United States, he ran out of emergency rooms. If you're under 18 and they can't figure out what you have and you keep coming back, they say the mom poisoned them, right? It's Munchausen by proxy. Second you turn 18, the kid's now a drug user, drug seeking. That's very, very unfortunate, but it's very true in a lot of situations. Did the genetic testing on him, found mutations in the calcium channel RYR2, 3 known mutations in this case. Put him on propranolol, and his disease he went into remission. He's also been off of propranolol multiple times, mostly because of his social issues, and the disease comes back each time. And then we put him back on propranolol, and it gets better again. And let's just go through some of these things. This is just showing you some of the cases I've shown you so far have been channelopathies. People say, well, this is mitotalk, what's going on here? There's a lot of connections between channels and energy metabolism. What happens usually is that the channels are leaking, and the channels are leaking a salt like sodium or potassium or calcium, leaks across inappropriately where it's not supposed to be. So that causes 2 things. First of all, the neurons are hyperactive. So the neurons are firing way too often because it's leaking. Instead of coming across when it's supposed to, it just leaks across. That causes neuronal hyperactivity. So you get things like chronic pain, nausea, ADHD, anxiety, you cause migraine, you get over-arousal of the neurological system. The other thing is that the body's going to try to move all that back, right? It leaks across, it's going to pump it back. And as it pumps it back, it requires ATP, it uses the pumps constantly. So people with channelopathies, it's not necessarily that they have a mitochondrial disease because they don't make enough energy. They have a mitochondrial problem because they use up too much energy. And again, as I said at the very beginning, most people don't have one problem. I'm trying to make it simplistic. But most of the people with channelopathies that have a mitochondrial-like picture also have a mitochondrial abnormalities as well. So so they make less energy and they use it more frequently. And so it's a combination that often causes issues. And then this is, she had really very, very severe dizziness, so she couldn't even walk. And she also had an RY2 calcium channelopathy, and then was placed on treatment and got better. It's just to show what I'm a science fiction geek or whatever. So, and that's my daughter when she was much younger. Just to go over this really quickly. Pam has autism, also has a few other issues, irritability, aggressive, OCD, the usual type of things. Some functional problems, easy fatigue, constipation. The big thing is that when you're doing this, it's not like you probably, those of you that have had genetic analysis before, it's not like the test comes back positive or negative. It's very, very complicated, and that when we actually look, when we get exome, we get about 30,000 different variants that we have to look at. We do, and that totally ties up the computer, makes it crash. When we do genome, we get 50 times that information, and then we have to use a server, like a Google server or something. We're working from that. But it's identifying which ones are likely to be related to disease. It's difficult. It requires the physician that knows the patient, that knows the genetics, and the family working together. This is a situation like when I found 3 different variants in this patient, and I put them in the order that I thought that they would be likely to be related: a sodium channel, potassium channel, and a creatine transporter. But after talking to the family, okay, we had a little bit for a while. What happened here?
There.
Okay. That it fit. Let me see this. Where is it? Okay, I'm not going to go back and try to find the slide that I'm missing. It just seems to not like it. What it was is it was to show that all the different possibilities on there, and how it turned out that this 18-year-old fit the creatine transport disorder completely, and that his personality traits and his issues were a very good match for that. And that was only figured out with talking to the family and directly about each and every one of them. They figured out it's likely to be a problem with the creatine transporter. He was then put on cyclocreatine, which is a type of creatine which can get into the brain without the transporter, and he improved dramatically. So the point, and then here, some of these. I'm going to go for that. So the point there was that you really need to have the physician sit down with the family and discuss the variants in detail and go over that to really make the connection. And that it's not the laboratory saying as to what you have. Many of you probably already have DNA tests that have been done, and they may have been negative. But it's to take somebody who knows that disease and to look at it and to go over it and, with you directly to see which other variants there might be. The other thing is that when you're talking about variants, you can talk about variants can be very common in the population, like we talk about variants that are 1% of people or even more common. They can be very rare. They can be so rare they've never been seen before. It may even be a new mutation that's not in the parents. You can have rare variants that cause disease. It's the single cause of the disease, or at least the lion's share of it. But the vast majority of variants, we're talking about risk factors, is that they contribute towards the disease, but they don't cause it by itself. And so you get risk factors from the mother, risk factors from the father. The kid has certain combinations of those risk factors, and then there's the environment. And the interaction between all of those contributes towards disease. It's not like a diagnosis as to what you have. Which is part of the problem. People are trying to look at this too simplistically, and they're not finding it because there isn't any diagnosis defined in most cases. So the thing here is about, if you've been to my talks before, mitochondrial DNA is still very important. Mitochondrial DNA is, the red ones there, is inherited from a female to all of her children. and then her females will spread it down. Those maternal inheritance of mitochondrial DNA changes are still very important. A lot of these families have shown you have that. But the thing is that everyone in the family is often affected to a different degree. This is one family of a mitochondrial DNA mutation which has been identified. Everybody in here will have like migraine or cyclic vomiting or depression or seizure disorder or GI problems or ADHD or some sort of other different problem. And then there's some people that really don't seem to be affected at all. Some are very severely affected. There's a couple kids in here that died, and other people just have migraine headaches. And the difference between it is are the nuclear variants. Is that some people have nuclear variants and some people don't. In this particular family here, the difference is the people that are severely affected have a TRAP1 variant, more severe than the TRAP1 family I've shown you before. This is one that destroys the TRAP1 gene entirely. And the ones that are lesser affected are negative for TRAP1. Talking about functional disease, this is something that, mitochondrial disease can cause kidney problems, liver problems, seizure disorders, all sorts of things. I just focus on the functional disorders because they're rare. Because they're not rare, they're actually very common, and quite frankly, because most other doctors don't. Things like chronic fatigue, chronic pain, GI issues, dysautonomia, mood disorders like depression and anxiety. These are all extremely common problems. The neurological system is electrical, it requires a lot of energy, and so it makes sense you'd have neurological disease, and a lot of what the nervous system does are higher functions about symptoms and the ways that you think about them. And here's just another slide that goes over many of those. Just go through these. I'll skip this one. This is one in which we actually did find a disorder of amino acid metabolism, which goes into the Krebs cycle. And then you can see here that it goes to alpha-ketoglutarate. The defect is between these two amino acids, but glutamate goes to alpha-ketoglutarate. We gave her a huge amount of alpha-ketoglutarate and she got a lot better. Okay. If you wanted to know what the sequencer looks like, this is an Illumina high-seq. The Human Genome Project you might have heard about, cost billions of dollars, and the world scientists were sequencing one person's DNA, and it took them 10 years. This machine can do it in an hour. Wow. When you're talking about DNA sequencing, you're talking about doing parts of genes, to single gene, to small panels like 20 genes that cause the trouble, large panels, maybe a few hundred genes. Whole exome is you do all 23,000 genes, all the genes in the body, and then we're now up to whole genome, where in addition to all 23,000 genes, you do all the rest of the DNA that's not part of it. Where we are really kind of depends on patients with functional disease. I'm mostly doing whole exome now because it's cheaper, and we usually get something treatable in the whole exome. And if they have some very severe autistic problem or something like that, we usually go to whole genome because this gives us a lot more information. In particular, this gives us information that you would normally get from a microarray, large mutations, you get Fragile X, you get a lot of different things from it. So to some extent, it depends on what insurance will pay for and the issues in the family. What does the DNA analysis do? It will give it right therapy. Many times the right treatment is not a drug. It can be cyclocreatine or alpha-ketoglutarate or vitamin C or something like that. Sometimes it's that we do find a therapy that's related to the molecular problem in the patient that's not a drug, but many times it is a drug. It may be Kytrol, it may be propranolol or something from or it may be acetazolamide. But instead of just trying different drugs and seeing which one works, is that we can actually figure out which drug it is that would be helpful. And then if you decide the drug is the right dose, some people are ultra metabolizers. They metabolize it so fast you have to give very high amounts of the drug. So if you just gave the normal amount and it didn't work, you don't know if that drug worked or not. The liver just got rid of it immediately. Some people are hypo-metabolizers. They don't metabolize it very well. So a normal starting dose can be very toxic. And so you get side effects and you stop it, but that may have been the perfect drug. Maybe start at a quarter of the starting dose, and maybe that's all that you need. So it also sometimes gives us information as whether the drug would be useful or not. So whether you need a drug, which drug, and which dose, these are the type of things that DNA testing can give. I'm not even going to go into this, is whether you test the parents or not is very complicated. In general, if you have a very severely affected child and mild or not affected parents, it's often a de novo or a new mutation, so you have to look at both parents to see what in the child is new that wasn't there in the parents. And if you have the whole family is affected or you have milder disease, then de novo is very rare and you can get away with sequencing the child. I have recommendations for testing. I don't really have time to go into this. For people with neurodevelopmental disorders like epilepsy, autism, intellectual disability, and then also ones for like complex functional diseases, the ones that have the weird migraines or cyclic vomiting or chronic fatigue or GI type things. And you can see that, like whole exome and mitochondrial DNA, singleton, just sequencing the child, and then the expert interpretation. Even if you had it negative before, you really want a doctor that can go over that with you. The costs, I mean, obviously this is a thing. When I was working for Cortigen just 4 years ago, we did 3,000 genes, and it was $30,000. And a lot of you had that testing. That was amazing at the time because only a couple of years before that it was $5,000 a gene, and it took a year to get results back. So when we can do 3,000 genes, we can do everything in the mitochondria plus a lot of other things for $30,000. It was like a miracle type thing. Of course, now we can do all of the genes, all 23,000 genes for $1,000. So it's changed a lot. There's interpretation costs on that. There's, like, you get an MRI, you pay the hospital for the MRI, then you pay the radiologist to read it. But the sequencing now is pretty much automated, and you can get tremendous amounts of data very quickly. The difficulty is in the interpretation. But the price are now 10,000 times less per nucleotide than they were just 4 years ago. You say, well, if I wait another couple of years, they're going to pay me for it, right? And it's the way things are going down. They came down and now they're starting to come up again. Not a lot, but we've pretty much hit rock bottom because it's just the collection and everything, and it's kind of much hit rock bottom. Prices are starting to come up a bit, as a little bit as laboratories find they can't do it for those low prices. But I think that it's at a good time to do it right now because you can get all of the testing done, and it goes on the computer. Is that it's on a computer, and later if you have any questions, there's the sequence there, it's already done. A lot of times people will ask me, well, what about this gene or that gene, and I have it on my laptop. And I can send, if you go through me, I can send you an Excel file that has your entire exome on it and everything on it. So if you go to any other doctor at any time, it's an investment in the future to have that sort of information. And you could do the whole exome plus mitochondrial DNA plus interpretation for prices are around $2,400. That's cash price. If you go for insurance, insurance could pay some or all of it. Just to warn people, the insurance company will pay a lot more than that because the companies raise their prices about tenfold when it's insurance. Because 90% of the insurance companies won't pay their bills, so they have to skip the ones that do. I know that sounds bad, but it's reality. It's the reality of the system that we are forced to live with. This is an Excel file. You can see that this is an alphabetical order of the genes, the variants, the change in the genetic code on the nucleotide level, the DNA level, the protein level, and then the color codings are, I told the computer where to put the colors on there so they'd be interested. This is somebody with autism. Actually, this is Dr. Temple Grandin, her sequence that I've been working with her on. This is just a random part. This is the very beginning of the sequence. These are the ones that I think are actually related to her disease. In addition to finding out several things that are likely causing her high-functioning autism, we also found several treatable conditions that she has to take care of a lot of the symptoms that she has. And then this is just, if you think that the medical team looks at you like this, that's the poisoner, which is unfortunately very much the case all throughout the country here, including some cases locally. is that, you know, this is what the legal way of looking at it and the medical way of looking at it, you know, that unfortunately the complexity of what's going on is really, is way beyond the training and understanding of most of the people in the medical field. And they really aren't up to date on the latest science. So that there are, I did give a talk to MitoAction that is archived, right? Yeah. On this, so if anyone feels that they may be, people might be looking at them a little funny, you may want to listen to that. The main reason to do that is to find out where are the buttons that you may be pushing that you don't even know about, that may be causing people to say, I wonder, is she doing that? And I'm sorry, but it's almost always the moms that are being identified. And there are many, because I hear about these cases all the time and I go back and talk to the people, I know what the buttons are that makes the team think about those sort of things and what leads them to think about it and what leads them to actually file. And it's most of the time it's not anything that the family or the mother did wrong. It's just certain things that are done that aren't necessarily, so if you know what to avoid in what sort of situations, then you can try to avoid this problem, and if you start to go down that road even a little bit, you can reverse course and go in the other direction. It's very rare that someone says, this is it, and then they start filing. It's usually there's several things along the road that cause them, so you can reverse course on that. And a lot of the things that I've been talking about have to do with antioxidants, because there's in mitochondrial dysfunction, whether you get there because you're using energy too quickly, or you're not making enough energy, or any other reason you have an oxidative state, and we want to have antioxidants on board to try to work with that. There's several different things that are neuroprotective that are often helpful, like magnesium and zinc, and then there's things like creatine or alpha-ketoglutarate that are helpful in certain situations and don't harm anybody. So what I was doing is I was putting all the stuff together into one. So I have my own product that I work with a bunch of people, and you can see there's one other guy you probably recognize up on there, Dr. Frye. It's been helping me put this together, and we have some up in the back there if you want to do a taste test to see if you like it, and I can also sell a few of them today if anyone wants to do that. You can buy them online. And that's it.
I wasn't cutting you off, the sleep timer was getting ready to go.
Okay, oh, I get it. Yeah.
Would you let everybody know what is the best thing if they feel they've either had the whole exome sequencing that was negative, what would be the next best step, or what is the next step if they want to do that testing? Regarding, like, what you say, get the raw data, call collectibles, or whoever you want to interpret it, that type of thing. Let people know the next steps.
There are some places to go from there. That you can go up to whole genome. One thing to do would be to go from whole exome to whole genome. You do find more things on genome that you do on exome, but it's not like, even though it's 50 times larger in sequencing, you don't find 50 times more. You might find 25% more, because most of the stuff is in the exome, but that's one thing that can be done. You can also sequence things that haven't been done before if you haven't looked at large mutations, if you haven't looked at mitochondrial DNA, maybe Angelman, something that needs to be looked at. So there's a possibility that there's more sequencing. But like you alluded to, often it's on the exome. A lot of people have exome, they have mitochondrial DNA sequencing. My expertise are the functional disorders, pain, fatigue, depression, all that sort of stuff. Plus I do the autism and other neurodevelopmental disorders. So it's in one of those areas. I'm very good at going through the DNA and finding things that are treatable.
So do you think that the whole genome sequence is better for somebody who says nonverbal for autism?
If they're nonverbal and the parents are unaffected?
Unaffected, yeah.
Yeah, then I would say trio whole genome. Then you're talking about some money in that situation.
So that'd be more.
But like I usually go for variant YX. Again, I don't have any connection with any of these other than I ordered from them. It's a good company and they have a good price. They will do the cash price is now, it was lower, but they raised it because they were losing money. It's $2,800. So if you did, but you double that, not triple that when you do the trio. So it's $5,600 plus there's another interpretation charge, so there's $6,600. That's if you paid cash. That's worst case scenario.
Sure.
I'd say 2/3 of families are getting it paid for by insurance.
Well, I'm sorry, what's the name of that price?
It's variant YX,
Thank you.
but those aren't their general prices. That's if I'm involved in it, you can get those prices. There are other prices, or I think twice that at least.
So what's the best way for people to reach out to you if they want to?
Just my email address.
Okay.
Yeah, I have that on there. I have a lot of different emails because I work for different things, but if they wanted to, if you want to talk about having me be the physician and to see the child either telemedicine or in person in Pasadena, this is Dr. Boles at molecularmito.com. If you're interested in me helping your doctor to read the DNA sequence and have your doctor do it, then you would contact neurogenomics at cnnh.org. And then this is for the neuro needs formula, the special needs back there. Yes?
Okay, because when I noticed your presentation and stuff, you were talking about a lot about GIs, and you were talking about that patient that had the chronic migraine, and you were saying that it was trap 1 or something that they found, or?
Well, there was a variant or a change in the genetic code in the TRAP1 gene that
So
I
how do you-
found
okay.
in that. I'm sorry?
Okay, so how do you get tested for something like that? Because
If you get a standard test, actually, if you get a test anywhere, they won't find that because it's 1% of the population has that, and it'll get filtered out by the computers. There are literally about 20,000 different variants that all of us have that are at the 1% level or more, or so.
yeah, 'cause
So
there's-
those are filtered out. You have to actually know what you're looking for. What I do is I take a list of there's a few hundred genes on that list, and I look at every variant in those few hundred genes because those are common causes of problems that look mito-y, and they're also treatable. And then I look at the other 23,000 genes or so to see if there's anything that I can do about it. But you have to, I look for that variant specifically on that. Yeah, I found one just last week on somebody that fit perfectly. That had gone through.
Yeah, because he's got with our son, they're sending us pain management for abdominal pain and things like that. That he's been getting to the point that he was just in the ER just a few days ago because of it. So we're sending him for pain management, but I don't know if anybody's ever determined he had that gene or person.
No, nobody would have done that. I mean, there are things, TRAP1 and other chaperone type things can do that. Channelopathy is really common. Channelopathy is even more common. Sodium 9A, 10A, 11A cause chronic pain problems. Several of the calcium channelopathies, many of the neurotransmitter diseases, and then there's the axonal transport disorders that can also do that. Plus there's many, many others. So it really, it requires looking at all the variants and all of the genes and knowing what you're looking at.
I just wanted to share that for most people here who goes to a physician locally, maybe your pediatrician or whoever, order the mainstream labs, just so you can know from a personal experience, we went through a very well-respected mainstream lab and ours came back negative, no findings, and I was able to get the raw data, and once that was rerun through Dr. Boles, then he was able to pick out the ones that were specific to our functional issues, and it made a huge turnaround for our kids. So just because you think, oh, the doctor says it's a good lab, unfortunately, they're not looking at these specific genes. And so that's what I was trying to express to everybody here, that they need to know that, because even if they tell you, well, the whole exome was negative, we didn't get anything out of it. Try to, if you really feel that this is pressing, you should take it to the next step and obtain that raw data or go further testing so that you can try to get, some type of answers. You're not going to have just one gene that he's safe. You have multiple things that he can help get you focused on to help get you or your child better.
I mean, there is so much in the exome. It's just, it's getting in trouble. I won't crash it for you. It's starting to have trouble. But it's that one thing I showed you, there's 40 changes in the exome that's related to Dr. Grandin's disease. That are potentially. There's so much information there. Yes.
Yes, thank you for coming and visiting. It's a nice presentation, and also we learned. And one point, like, the de novo mutations are very high in neurodevelopmental disorder. And what might be the cause, or what do you think? And I have another question also.
Oh, I mean, the de novo mutations tend to be I'd say half of them are transcriptional factors, which are hard to treat. That's true, but at least you have a primary diagnosis and know what's going on. But many of them are in genes that are treatable. Every once in a while, I found one that was in TRAP1 once. So you can find them in treatable genes sometimes, and even if you do have a de novo and a transcriptional factor and you can't treat it, you can have modifiers in other genes that are not de novo that are treatable. So sometimes the primary mutation, there really isn't something with current technology you can do anything about, but that sometimes you look for modifiers to be treated. So I'd say that synaptic regulators, any of the genes which form the synapses or that migrate the nerve cells to make synapses or actually hold the synapse together, those are very commonly de novo as well. So,
yeah, another one, like, there are some epigenetic phenomenon also working. It's non-coding, and how does it helps in long run?
There's got to be something to do 5 years from now, right?
Thanks.
We don't have, though that there are some things like Angelman and Prader-Willi which are kind of involved in epigenetics. There's really not the ability to clinically look at that now. That's a research question. But maybe another 5 or 10 years we'll have an epigenetic test for $1,000. I hope so.
Yeah,
but I can't. Right now, it's just one of those things that you do have. Occasionally, there's a patient that doesn't have a variant that I can find related to disease, or that has a variant that may or may not, and nothing works on the treatment. Maybe 20% or so. It's pretty unlikely. It used to be 95% of my patients just 10 years ago were undiagnosed. 95%. Now it's like 20%.
So, yeah.
So, made a big difference.
Thanks.
So I saw them up for you.
All right. Sounds like you've solved most of the problems.
I didn't talk about that scope after therapy. That's your job. Oh yeah, I didn't know what you were.
How can we see it?
Just email me.
Email you?
Right now.
And will we have access to your slides too?
There MitoAction has it.
Okay, perfect, thank you.
So now for something completely different. So I actually have the pleasure of meeting Dr. Boles almost a decade ago now, and there we go. And it was really interesting because I had just been really looking into the connection between mitochondrial disorders and autism, and still it's kind of a field that's really very evolving, and it was hard to convince people that there was actually a connection there. And I had met Dr. Boles. I was actually before I went to Arkansas. So I've just come here to Phoenix Children's Hospital. I've been here about a year. I was at Arkansas Children's Hospital for about 6.5 years, and before that in Houston. And just before I went to Arkansas, I was interviewing at LA Children's. And I don't know if you remember when I met you, and Dr. Boles was the chief of genetics at that time at LA Children's. And, he was a geneticist, and most geneticists think everything's genetic and you can't solve it, at least a lot of them do. So I was very skeptical. I said, I don't know how much I should talk to this guy about mitochondrial disorders and how you can treat kids with autism, because a lot of people don't believe it. So, I threw out a couple of things about how, we see these and of course, as soon as I said that, Dr. Boles goes, they all have mitochondrial disorders. Oh, I treat them all. And so, we, I think, bonded at that point. you know, because we kind of understood that there was something more going on than just simple genetics, that the body actually takes the genes and makes the machinery work, and sometimes the machinery doesn't work very well, and you have to figure out why it isn't working and what you can do about it. And many times you can do something about it. And you know, as Dr. Boles has given us a lot of examples of what you can once you figure out what some of the gene variants are. So my specialty is autism, and I'm going to talk a little bit about the connection between mitochondrial dysfunction and autism. As Dr. Boles also had said, I hope there's no FDA plans here. Any type of treatments that I talk about today are not FDA approved. They're considered off-label, so in other words, don't try this at home. So make sure that any type of treatment is done in conjunction with a medical professional. So many people may know what autism is, they may not, so I'll just quickly go over how you define this thing that we know as autism, which has become more and more well known in our society because of the increased prevalence. When it was first defined, it was really thought of as when three different deficits kind of come together in children, and that is social deficits, the inability to communicate, and certain types of behaviors, what we call repetitive behaviors and restrictive behaviors. And when those three come together, we see something that we define as autism. We used to have these different categories, Asperger's, PDD, NOS, which confused a lot of people. And I think because it was so confusing, we have now, in what we call the DSM-5, the new diagnostic manual, have just made a big spectrum and called it autism spectrum disorders. And because social, the ability to engage socially is so engaged and so intertwined with communication, we don't actually separate them anymore. And so we say there's social communication problems along with these types of behaviors, these restricted interest and repetitive behaviors. And if you don't have the behaviors, then you can also have something called social communication disorder, which is separate from autism spectrum disorders. And one of the things I like to just emphasize with autism and to help understand it is, because a lot of people say, well, how do you differentiate a lot of kids that have language delay from kids that have autism? And because many children with autism have language delays, but in autism, it is the inability to communicate. It's not specifically language. Language is verbal communication. And so kiddos with autism cannot communicate. And that is one of the prototype missing pieces of behavior that we see with children. Something I think that's really underappreciated is the ability to point. We kind of take it for granted that we point to things that we're interested in. But this ability to point, which develops around 8 to 10 months of age, is really one of the most important social gestures we'll ever have. And this is something that children with autism do not develop. And this is your major non-verbal way of communicating socially. Just other things that other kids early on with autism, we see that there's other types of behaviors, which many parents talk about, including not responding to their name. Many times they go for hearing tests because they think the child can hear, looking at other people, showing objects. Showing is another thing I think that's underappreciated. If you have a little child, one of the first things they do when they can walk, or you go over to them as they're playing with their toy, and one of the first things they'll do is they'll take it up and they'll show you it. They'll hold it up and show you what they have. Children with autism don't do that. That's one of the major social gestures that's really important in development. So what causes autism? And when I started working with kids with autism, that was really the question. One, what causes it, and what can you do about it? And there's a big push or lots of talk about the genetics of autism. And the reason why that is because we know that autism has a very large heritability component. That is that if you have a child with autism, it's about 10 times more likely that you'll have another child with autism.
twins are more likely to have autism than siblings and such. And so these are ways you really look for genetic factors. However, what we found is that as we look for genetic factors and as we've been able to sequence the whole, everybody's genome and such, the fact that there's genetic factors there has not really laid to bear for most kids with autism. And these are actually even old numbers. If we look for very simple genetic abnormalities, such as cytogenetic abnormalities, that is problems with the chromosome, Fragile X, Rett syndrome, or chromosomal microarray abnormalities, that is copy number variations, where you may have a little extra piece of your chromosome or a little piece missing. And we can add in there now some rare variants. We find that for the most part, it really adds up to less than 20%. And in fact, the newer studies put it more down, 13, 14%. So what does that say? Is genetics not involved in autism? Because we just heard this wonderful talk about genetics and how it's involved in autism. And what I like to say is that is the fact that the science has lagged behind the technology and to a significant extent. That is that we're able to sequence a whole genome, but we can't interpret it very well. And that the genetics are not as simple. One of the things that we thought in the 90s is that we found many genetic disorders, and they were really single gene disorders. And the idea was we were going to sequence the whole genome, and we were going to find all these single gene disorders. And what we found is that's not the case. It doesn't mean that the genes aren't involved. It means there's really complicated interactions between genes. There may be variants that are not rare, common variants that interact with other genes and also with the environment. And so one of the things that we know about autism is that there's a very high genetic environmental interaction, and probably each of those components contribute about the same amount to the development of autism. And so in comes the mitochondria. You thought I forgot about it. So the mitochondria, I think, is the perfect organelle to study for autism, because it is very much affected by environmental interactions. So things like, and when we talk about environmental interactions, it's not just things people think of as like toxins, but we're also talking about nutrition. We're talking about our microbiome, our gut bacteria. We're talking about inflammation, different types of infections. All these things really affect our body. And then it affects this part of the body that's really important, that's in every cell in our body, the mitochondria. So every cell in our body has somewhere between, anywhere between hundreds and tens of thousands of mitochondria. And we think of it as the powerhouse of the cell. And there was one slide I think you skipped where it showed where the mitochondria was and where all the other metabolic pathways were. And it's in the center there. So when other disruptions happen in the cell, many times they'll affect the mitochondria and how it functions. And when the mitochondria doesn't work, many times it'll affect all those other pathways. So it's very, very important. Really, one of the reasons why we're talking about it is a little bit mysterious is it's really a new disease. And not unlike autism. That is, the first time mitochondrial diseases were described was in 1988. In the same year, two scientists, clinician scientists, described what we know as kind of the first mitochondrial diseases that involved mitochondrial mutations. We recognize there were diseases that which may have involved the mitochondria, but we didn't know that it was the primary culprit until really 1988. And in medicine, that's really a blink of the eye. So we know that individuals with mitochondrial disease have problems with clinical symptoms in very highly energy-dependent parts of our body, and that includes the nervous system, the GI system, and the immune system. And so if you're familiar with autism, you know that kind of recapitulates some of the what we see. And not only in autism, but other kind of mysterious diseases that we still haven't figured out. It's not just the powerhouse of the cell, but it has other functions. We know that there's this thing called mitochondrial disease, but then what we're realizing is that there may be sometimes where the mitochondria just isn't working right. It may not be the primary culprit, but because it's attached to everything else, that it may not be working very well, and it may be key in the network of abnormalities in the cells that are causing diseases. And we've realized that it's in a wide variety of different diseases: GI motility disorders, multiple sclerosis, unexplained liver failure, kidney disease, heart failure, epilepsy, psychiatric disorders, cancers, diabetes, and many types of learning disorders, cerebral palsy, autism, and learning disorders. So the evidence that the mitochondria was involved in autism actually goes back even before mitochondrial disease was well defined. Actually, Dr. Coleman, back in 1985, really wrote the first paper that really gave us some clue to what was happening. And she found that about 5% of the patients that she saw with autism had lactic acidemia, and elevated in lactic acid is one of the key biomarkers we look for to let us know that the mitochondria isn't working. And she proposed that there was a problem, a deficit in carbohydrate metabolism and pyruvate dehydrogenase, which is a key enzyme in the mitochondria. Back in 2012, myself and my colleague, Dr. Rossignol, had noticed that there was all these studies. There's a lot of studies talking about mitochondrial dysfunction and autism. So we did something that scientists do, is we said, well, what's the evidence? If we look at, are these just kind of random papers that are, studies that are of low quality, or is there actually some solid evidence? So we did something called a systematic review, where we looked for every paper that was published on this subject in the literature, in the medical literature, and then did something called a meta-analysis, where we put together all of the data from all the different papers and asked, what does it show? And is it really pointing in one direction or the other? And we found out, I'm not going to go over all of the findings, but one of the key findings we found is that there's actually 3 really good studies for looking for what we call classically defined mitochondrial disease in autism. And one of them was very, very, very well done. Screened every child in Portugal and the Azores for autism, and then for mitochondrial disease, and then looked for a diagnosis. And overall, when we looked at these studies, they said that about 5% of children with autism have what we call classically defined mitochondrial disease. And that's kind of a very tight box that you put around, that you say that this is definitely primarily a disease of the mitochondria. And that's a really gigantic number, because mitochondrial disease really affects less than 0.1% of the general population. So for 5% of any specific disorder to have a diagnosis of mitochondrial disease is really pretty gigantic. But then we noticed something else. When we looked at studies that looked for biomarkers of mitochondrial dysfunction, that is, that the mitochondria wasn't working well, we found that there was many studies. 6 studies looked for elevated lactates, were one of the main biomarkers of mitochondrial dysfunction. And overall, if you put them together, 31%, they found that 31% of children with autism seem to have an elevated lactate. And it wasn't just that, it was elevated pyruvates, 14%, elevated lactate to pyruvate ratio, again around 30%, and so on. And then you could argue that some of these studies used normative values instead of contemporaneous controls, but down on the bottom we actually looked at the studies that did use contemporaneous controls, and they also verified this. So it was one of the mysteries was, well, we know that 5% seem to have this classic mitochondrial disease, but somewhere around 30% have markers that the mitochondria isn't working very well. And then there's another study that I did when I was at Arkansas, where we screened, there's actually our data from a clinical sample from Houston, because you can say, well, many people say, well, you draw a lactate and sometimes it can be spuriously elevated because it wasn't drawn right. So we looked at the number of times that you can actually repeat this in patients, and we found that about most of the time, about half the time, these biomarkers do repeat if you test them twice. So it's not just a matter of the lab not drawing the lab correctly. And then you have this other data from UC Davis, the Mind Institute, where they looked at immune cells of children with autism. And they found that when they compared immune cells in children with autism to those in age and gender matched controls, that 80% of the immune cells had abnormal mitochondrial function. So that's really a gigantic number. There's been other studies that look for specific types of symptoms that might be associated with mitochondrial disease and autism, and we know that children with mitochondrial disease and autism in this study, this Weissman study, this was out of Johns Hopkins, show that there's high rates of GI symptoms, fatigability, and multiple regressions, and including late regression, usually not always seen in autism. In our meta-analysis, going back to that, we asked the same question, and we found that kiddos with mitochondrial disease and autism actually had specific symptoms that were more likely. They were more likely to have seizures, GI abnormalities, and motor delays. So motor delay is a big red flag. Because, a lot of times people don't differentiate hypotonia, which is a change in tone, versus motor delays. And it's unusual for a kid with autism to have motor problems, but it's a big red flag if they do have also delays in motor development, that they could have a mitochondrial disease. One of the big things that we also found that was interesting is that of all the cases of classically defined mitochondrial disease and autism, that only 23% of them, you can see down here, they actually found any type of DNA abnormality that could explain the mitochondrial disease. So it leaves open that, there might be other things going on. And then the story gets even more complex. And this was one of the first studies to really show this. This is a really neat study that was out of UCSD. And they found that there was a girl that had something called Lee's disease, mitochondrial disease, and she had a specific mitochondrial mutation, and the heteroplasmy in her of this mitochondrial mutation was about 80%. So 80%, you can think of 80% of the DNA or 80% of her mitochondria were affected. They found that when they looked at how her mitochondria was working, that both complex 4 and complex 5 were not working very well. And that's how many times you define classic mitochondrial disease. That is, the mitochondria is not working very well. It's really somewhere about 20% of normal. Okay? But then they looked at her brother. Her brother had autism. And actually, he had the same mutation, but it was only at 60% instead of 80%. And when they looked at how his mitochondria was working, actually, they found that complex 1 was sky high. It was working overtime for some reason. So instead of having a mitochondrial deficit in how the enzymes are working, actually the enzymes were working really, 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 really hard for some reason. And this is one of the first case reports of really looking at kind of this overactivity. And then when I first started seeing kiddos with autism, they fit into this box of mitochondrial disease. Sometimes we do muscle biopsies. And I started noticing that some of the kids that came in, we did a muscle biopsy, and we saw a similar pattern, not in complex 1, but complex 4 was actually about 200% of normal. And this is the first case report of 5 patients with complex 4 overactivity that we published. And it ends up that we're not the only ones. This is a study looking at calcium homeostasis and looking at brain tissue, postmortem brain tissue, and activity of enzymes. And they actually showed that also in their sample of individuals with autism, complex 4 was much higher than controls. One of the ways that we've gone on to study kiddos with autism without doing lots of muscle biopsies is that we started to use something called the Buccal Swab technique. And this is one of the studies that first showed that there is a reliability with it. This is something that was developed by Michael Goldenthal at St. Christopher's Hospital, associated with Drexel University in Philadelphia. He showed that the Buccal Swab technique, that you can look and measure complex 1 and complex 4 pretty reliably. So we've looked at kiddos with autism and used it to screen for mitochondrial dysfunction. And this is some of the data of one of the papers we published. And what you can see is that overall, that if you look at complex 1 activity, this is corrected for citrate synthase activity, you can see what happens is that actually the activity overall in all these kids, these are all kids with autism, there's an increase in the variation of mitochondrial function. You have many, the red dots on the bottom here, show that there's lots of kids where complex 1 is not working very well, it's outside the normal range, and there's other kids where complex 1 is way above normal. And we see this in complex 4 too, but it tends to be way above normal. We also did something called the Vineland Adaptive Behavior Scale on many of the kids that came in, and we correlated it with mitochondrial function. And what we found is that if it's too high or too low, so both ends of the spectrum was associated with lower development. Interesting other studies that we've done is, this is a nice study in Phelan-McDermott syndrome. So again, when you, this is probably the disorder that's the genetic disorder that's most closely linked with autism. It's really thought of that there's one gene on the chromosome 22q13 called SHANK3 that is responsible for the symptoms, the autism-like symptoms. However, what we did is we actually looked at that area of the gene, and we found out there's actually 6 mitochondrial genes also in that area. So when you have a genetic syndrome, you know, this is an example of one you might be able to do something about if these genes that influence mitochondrial function are affected. And so in our first study, we just wanted to say, is there any abnormalities in mitochondrial function in these kids, as you would predict? And the genes that I show you concentrate on complex 1 and complex 4. So our hypothesis was that complex 1 and complex 4 would be affected in these children, but not complex 2 or 3. And so we used the Buccal Swab technique. Dr. Goldenthal was actually able to also measure complex 2 and complex 2+3 activity. And that's exactly what we found, is that kids with Phelan-McDermott syndrome, that there's this widened spectrum of mitochondrial function, with some having very low complex 1 activity, some having very high complex 1 activity, and some having low complex 4 activity. One of the other things that came out of this, we looked at the kids with abnormal complex 1 activity, and of course we're very interested in kind of this overactive activity, and what we showed is that the kids that had overactive complex 1 versus underactive were more likely to have autism and more likely to have regressive type autism with the classic type features of autism. So just some data kind of pushing it more to suggest that there might be some connection between this. So we kind of think of, so we're starting to think of this idea of how the mitochondria and autism are related, is that if you look at autism spectrum disorders, there is definitely an overlap with mitochondrial disease, as you can see at the bottom, but there's also something to do with this kind of non-classic mitochondrial dysfunction that we can't quite understand now, but what's important is that it can be a treatment target, because, you know, if we know how to treat some mitochondrial disorders. One of the things, I'll just go quickly, I don't have that much time, of some of the research we did. We used this thing called the seahorse. The seahorse has really advanced our ability to measure mitochondrial function. In the past, measure mitochondrial function in the laboratory, you'd have to isolate individual mitochondria, and you only could do one sample at a time. The seahorse has this little tray here, which is a 96 well plate. So you can actually put 96 samples and measure mitochondrial function on 96 samples at the same time. And you can put living tissue in there. You don't have to isolate mitochondria. So it's really increased the ability to research a mitochondrial function. So, and what it does is it gives us these different measures of mitochondrial function, something called basal respiration, which tells us how the mitochondria is working, ATP respiration, which tells us how much ATP, that's the energy molecule, how much energy is being made, looking at something called proton leak, that's something that's used to regulate oxidative stress. When there's high oxidative stress in the mitochondria, kind of a safety valve, but makes the process inefficient. And then you can look at something called maximal respiratory capacity, that is, what is the maximal ability of the mitochondria to work if it really needs to work. And if you subtract that maximal versus the basal, you get something called reserve capacity. And this thing called reserve capacity is very important because it tells you how much backup, how much reserve the mitochondria has if it has to actually, if the cell's under stress and it has to produce energy. And we know if reserve goes to 0 or it needs to become negative, that the mitochondria in the cell are in trouble. So one of the things that we did in our first experiment is we first asked, well, you know, what's the difference between tissue? We used immune cells called lymphoblastoid cell lines from kids with autism and controls. And of course, knowing that there might be increased incidence of mitochondrial disease, we said, well, probably there's going to be, the mitochondria is going to be less able to make ATP and energy. And what we found, this was some of our first experiments, is we found the absolute opposite. That actually the cells were making more energy, more ATP. They were working overtime. So ATP-linked respiration was higher. Reserve capacity was higher too. And then what we did is we stressed the cells. We said, well, are they more vulnerable to stress? And what we found is that indeed they were. We used something called DMNQ in increasing doses to stress the cells, and we found out that the reserve capacity went from very high to very low, below the controls. Although if you look at the lines, you say, well, you know, that's just a tiny little bit. You know, what is that really significant? So, of course, everything in autism is subsets. So we asked, is there really a subset driving this? And we found that about a third of the cells really showed this phenotype of having this abnormal profile. And when we separated them out, we can see that the effect is more dramatic. That these are the cells that really show this abnormal mitochondrial function. And they're actually making twice, for some reason, just sitting around, they're making twice as much ATP for some reason. So they're really working overtime. Their reserve capacity over here is twice what the controls are. And when you stress them, you can see what happens is that reserve capacity goes down very, very quickly below the controls. So it says that they're more vulnerable, actually, to oxidative insults, physiological insults. You know, and you can think of that as inflammation, also environmental factors, also. So those were cell lines that we got from a biorepository, the NIH and some other biorepositories. So then we asked, well, what if you just carefully control and you look at siblings of kiddos with autism, their cell lines, versus the kiddos with autism and other controls. And what we did is we were able to replicate the same data and show that even compared to their sibling controls, what distinguished the kids with autism is they had this abnormal mitochondrial function with an increase in reserve capacity. Their mitochondria working overtime. And then what we also did, which was kind of impressive, I think, is we went back to the examinations that they did many years ago, probably decades before we did this experiment, on the kids when they took their blood. They did something called the ADOS, which is the gold standard diagnostic test, and they looked at repetitive, one of the scales is repetitive movements, and we actually showed that the kiddos that had the more extreme abnormality in mitochondrial function, that is, they were making more and more ATP, which is this red line up here, had more severe autism symptoms. So probably 10 years before we even did this experiment. So suggesting that this physiological abnormality was actually related to their behaviors. Okay, so then our kind of model of this is that, well, maybe something, it could be genetic or it could be environmental. We thought, well, maybe there might be some type of environmental insult that really caused some changes in the mitochondria to cause it to be in this state. So one of the things that we did is we did another experiment in the laboratory. So we said, well, what happens when you get exposed to adverse environmental situations such as inflammation, certain types of environmental toxins and such? One of the common themes is that there's an increase in oxidative stress. So we said, what if we took these cells and we exposed them to low levels of oxidative stress, that is an adverse physiological situation, simulating what would happen if you had prolonged inflammation or you're exposed to some type of environmental toxicant for 96 hours. Would this change the physiology of the cells? And what we found is that yes, it did. And if you look at this graph here, we can see this is the graph of the cells for the reserve capacity before in blue, and then after we exposed them, the reserve capacity went up, just like the abnormal cells that we found associated with autism. And we found that the maximal respiratory capacity, this is a little bit more detail, but we found that especially the subset of abnormally functioning cells with abnormal mitochondrial function were very vulnerable to those changes, but also the control cells also had some changes too, suggesting that this may be, a general reaction to cells with the mitochondria. There's other types of evidence for that too. We also looked at other, this is another experiment where we used something called chlorpyrifos, which is a toxin that's been associated with and linked to autism, and we found out that incubating for 96 hours with this toxicant at a specific concentration, you can see here, actually increased reserve capacity in the long term too. So this is some data that suggests that the environment may in fact be changing these cells this way. And then you can say, well, that's all great, everything happened in the laboratory, but what about patients? And so this is an experiment where we actually looked at kiddos with autism to see if they also had the same abnormality in reserve capacity, heightened reserve capacity. And we found out that in our sample, about 25% of the kiddos that volunteered for this study, this is from Arkansas when I was there, seemed to have an increase in reserve capacity, and you can see these in the red dots. And then when we took kiddos with classic mitochondrial disease, they did not have this. And that's this graph over here, so that we're suggesting that this is actually a type of mitochondrial function that's actually distinct from classical mitochondrial disease. And this is kind of exciting data that has just come out, where we've actually now looked at their exposure to air pollution, something called PM2.5, which has been in the news a lot because it's linked to many diseases. And we found that actually their exposure during gestation, so before they were born, is linked to changes in mitochondrial function when they're children. So what can you do about this? So this is where sometimes a controversy comes, and this is, I think, a paper which is very timely about mitochondrial dysfunction and whether you treat it or not and how you interpret it. I know the Mitochondrial Medicine Society just published a similar paper, so we were first though. Dr. Nizoff is a little bit mad about that. They didn't include him on the author list for the new paper. He said we were first, but I think it just shows that there's more and more recognition that there's something there, that those kiddos and adults that don't fit into the box of classic mitochondrial disease, we have to understand them, and we have to think about treating them. And in this paper, we talk about this fact that if you don't have primary mitochondrial disease, can it be secondary? Is that a real thing? And do you treat it? And actually, in this paper, we have a few cases where we talk about, disorders that are clearly secondary mitochondrial dysfunctions, such as Duchenne's muscular dystrophy, which we know the gene has nothing to do with the mitochondria, but the mitochondria is involved, and some of those kids get better if you soup up their mitochondria because it's involved in the disease process. Of course, there's all these cofactors that we use to treat mitochondrial disorders. What's the evidence? We have that it actually helps in autism. This is a study where we actually looked. This is kind of an observational study of kids that came through our clinic where we measured mitochondrial function. We found that there were certain supplements that seemed to improve their mitochondrial function, and we also split it into kids with mitochondrial disease and no mitochondrial disease. So complex 1, just by observation, we found out that fatty acids and folate seem to be helpful for citrate synthase, fatty acids, folate, and antioxidants. And then what we also did is we looked at not just the absolute activity of the enzymes in the mitochondria, but how they were working together. And we actually showed that those that took folate, there was a greater relationship, a tighter relationship between complex 1 and complex 4. And those that were on B12, there was a tighter relationship between complex 1 and citrate synthase activity with both folate and B12. So, we also have to think about how we look at these things too. This thing of absolute activity, where it is being below 20% or 200% may not be, something that may be too simplistic. We also have to look at how the different parts of the cell, how the different parts of the mitochondria are working together. So a little bit more. And this is really one of the studies that was done, that is being done right now, which I have to, really congratulate these guys, Dr. Goldenthal at St. Christopher's in Philly, where what they did is they actually found kids with autism and mitochondrial dysfunction, gave them a mitochondrial supplement, showed that the mitochondria activity got better and their behavioral symptoms improved, and then they took them off of it, and they showed that their behavior got worse and their mitochondria got worse. And so we're trying to do the follow-up to this study now with NeuroNeeds, the Spectrum Needs product, where we'll actually trying to show that kids with autism and mitochondrial function, that it's a thing, and that you can treat it and the kids get better. There's other studies too, I'll just mention, that really look at the mitochondria but use supplements that would affect the mitochondria. This one's from Jim Adams at ASU, who's done a few of these studies looking at vitamin mineral supplements that would really help the mitochondria. He showed that certain measures of mitochondrial function improved, certain measures of behavior improved. And then there's 2 studies on something called carnitine, which you probably all know about. We know that there's 2 double-blind placebo-controlled studies that show that core symptoms of autism improve with carnitine supplementation. And in this study, they actually showed that the increase in serum carnitine actually was related to the improvement in many of the symptoms. There's another study. So these things are complex. They involve many of the metabolic cycles. This is a nice paper we put together in 2014 that describes some of the other metabolic abnormalities and how they can be treated in autism and what the evidence is. And this one also looking at a wider variety of pathophysiology. And this is just the team that helped me with a lot of this research when I was in Arkansas. And we have some of the team here that's helping us, Dr. Singh and Dr. Kareem. You can raise your hands.
Thanks.
Who
For autism.
will be part of the new team to help us try and figure this out. So thank you so much for your attention. I really appreciate it.
Thank you, Dr. Frye and
Yeah.
Dr. Boles, for your time.
My pleasure. Thanks for inviting me.
I would like to invite you guys up for some for questions, right? Yes, okay. I was like, I know that they've answered so much right now. You guys are full of so much information they've just given you guys. But if you guys want to look for some questions from anybody who has some, well, here I have one over here.
I got a question, ma'am.
Sure.
About sometimes parents not knowing when their kids are going to have Mito or not. How is that always predicted?
Who would answer that one?
I'm not sure how to answer that.
Yeah, I mean, I think it's complex, you know, and it's important, you know, parents that know their children well and they bring them to their doctor and their doctors carefully listen to them and try and find out what's wrong with them. It might not be Mito, it might be a lot of different things, but it's just important to you know, to talk to your doctor. If you have a problem, it's important to talk to your parents, and it's important to talk to a doctor and find a doctor that listens to what you're saying.
Got it.
You know, and you know, some doctors are busy, and other ones tend to be to like to listen, and so it's important to be comfortable with the doctor that you're seeing.
Got it.
So Dr.
Go ahead.
Frye, so you are seeing pediatrics?
What's that?
Do you see pediatrics? Or
I
you
do.
see
I'm at
adults?
Phoenix Children's Hospital.
Is there someone in town who sees adults?
You know, I see all ages, but I don't really know. Do you know, Wendy, if anybody
For
sees
autism
adults?
specifically?
Autism or Mito?
Or Mito, sorry.
Mito.
Yeah, that's a great question. We're actually working on, so we're on a committee to work on improving transition to, and so we're sort of working on identifying in the valley who's interested in children with complexity. And so I would say that's coming, but I have like a list on my phone of people who might be good, but not specifically for Mito.
But, you know, most of us, if you can't find a doctor, I think we try and do our best to see adults also. Do you see adults?
I still have, you know, mid-20s in my practice too.
Yeah, and I definitely, you know, see adults that if they can't find other providers, then, you know, we try our best.
Also, you can go to MitoAction's website, and they can also refer you to doctors in your area.
And then I have one more question about the information you gave. When we were talking about folate, were you talking about methylfolate or regular folate?
Yeah, that's a whole nother lecture. Yeah. So a lot of part of my other research is on folates and reduced folates and right. So one of the things you're bringing up probably is folic acid that we think of that's fortified in our foods is the oxidized form of folate, which some of our bodies don't process that well. So usually reduced folates are more bioavailable to the body. And there's many different types of reduced folates that you can use depending on the situation.
But that would be the typical that was used in that study where they just looked at the kids who were taking B12 and folate.
Right, we did not distinguish of whether they were taking reduced folates or folic acid, but I would say that most of my patients, those are most of my patients, were taking reduced folates. Yeah.
Dr. Boles, for somebody with CDS who's basically morphed into like the abdominal migraine area and the chronic fatigue, and it has pretty much, you know, gone less and further away from the cyclical vomiting part of it. What kind of sequencing would you be looking at, focusing on?
Well, it's the same condition, and around 2/3 of people, somewhere around puberty or so, it kind of progresses. often to abdominal migraine or migraine headache, or sometimes a dizziness or something like that. But it's the same underlying genetics. There are maybe 10 genes which are common in that situation, and there's who knows how many genes are uncommon. I keep finding new ones. So what I usually do is, because even if we find the gene that's probably related to it, that doesn't mean we found other things that are treatable. So I usually recommend do whole exome sequencing and mitochondrial DNA.
Okay, and if somebody were to do that, and were to see you in Pasadena, would that be over like a one visit thing, or would that be?
Well, although I've gotten everyone under control that I've done DNA testing on, it's not always the first drug and the first dose. In fact, it rarely is. Usually we'll start on the cofactors, the mitococktail, while we'll get the DNA sequencing, because the cocktail works really well. Sometimes that's all you need. We'll tie up the cocktail, make sure that it's high dose and that the blood levels are adequate. And then we'll do DNA sequencing and see what's left of the disease after the cofactors and what the DNA sequencing shows. And then based on that, make the joint decision as to if the drug's going to be used and what drug and what dose, and then we go up on the drug and get blood levels, and then even if you have the perfect drug at the perfect dose, it takes 2 to 3 months before it gets better. Because the body needs to heal once you've turned off the problem, basically. So it's more involved in that. So although it's nice to come to the first visit in person, it's not absolutely necessary. It can be done telemedicine. It's nice to do that. The follow-up's going to be done by telemedicine. At least here in Arizona. Everyone here lives in Arizona, right?
Yeah.
For where?
Atlanta.
Oh, okay. I have a Florida license.
We're over 2,000 miles away. Glad you made the trip.
What's the process to being able to have our child see you?
Just email me.
Okay.
Yeah, if you email, of course you just email me, I can reply and say what I want, but the main things I just want to know is, the date of birth, the name, a paragraph or two as to what the situation is, and then, when you're available. That's the main thing.
And if they have a clinical diagnosis, do you still see those patients? Or they haven't had
There's so many different clinical diagnoses. Some are wrong, some are correct, some are, yes, it's correct, but there may be something else. So yeah, it doesn't, yeah. I think that you have over there.
Yeah, I was talking about the same thing, because when I noticed your presentation, I was thinking, was he tested for things? Because a lot of what I saw overlaps things that are with him, so I wanted to be able to get in contact with you again about things too. So he said to email you then?
Well, the email is really good for setting up appointments and, getting the documents back and forth. And it's also if I've already done a full evaluation, like you have a new symptom or something, just to run it by me. Is this a problem? Do I need to do something? But I really can't answer questions about specific diagnoses until I've seen the person, because it's not only illegal, but it's also quite dangerous, because if I don't know the whole situation, I could say something very wrong.
So does it cost to do the telemeds, or how does it all work with you then?
Yeah, the telemed, whether it's telemedicine or in person, it's all the same.
Okay.
In terms of the cost.
So does that does somebody need to refer?
It's just my costs are quite high, because I go through the chart, you send and I go through all of that, and then we talk on the phone or in person for 3 hours on the average, and then I write up the note, and then later there are issues. So it takes 5 or 6 hours to do something, and I do charge $1,200.
Okay. And then Dr. Frye, you said that you're at Children's Hospital. So how do you get to see you to talk about some of the things that you mentioned that? Now that's Buccal Swab. Is that some kind of test that you do?
The Buccal Swab, yeah. It's called. There's now it's gone from the research laboratory to a commercial product called a MitoSwab. And it's something that may be very helpful in looking for mitochondrial dysfunction. You can contact Phoenix Children's Hospital. I'm in Department of Neurology and Developmental Medicine, and they'll connect you with Jessica, who's my coordinator.
Okay.
If you have your primary care doc, send a referral.
Okay, so they send a referral.
Yep.
Okay.
May I ask something off topic? Just a tad bit. Basically, just quickly, I was a Schaffner patient who went through all the diagnosis process, and then my neurologist said, hey, we don't know that we believe this anymore. Years later, and then he disappeared. Curious your take, can you just give me a quick summary on, do you still believe what those diagnoses came up with?
Well, there's a lot to say about that. First of all, is I think that everyone in this room can understand that doctors do not necessarily agree with doctors. In fact, in an area where it's new and controversial, they almost never agree. I mean, and there's more good than bad. I know there's a bad side, I see that all the time in the families, but the good side is that people aren't just saying this is what it is and they're wrong. People are starting to question it. Maybe there'd be something else, and they're looking for more issues and things. So it makes it very hard on families, but at least we're not just set, this is the way it is, this is what you can do. We're always looking for new diagnosis, new treatments, new ways to help. So a lot of the things that were done years ago, they were great and they were state of the art, but I mean, what we can do now is at least 100,000 times to 1,000,000 times better than even 5 years ago. So the stuff that was done beforehand may not be relevant anyway.
Well, we had cerebral folate numbers that were low, and then people threw questions. So my other question would be based
Well,
on
that, you probably might say something about that. That is probably a related phenomena that a lot of people with mitochondrial dysfunction do have cerebral folate deficiency. It's not their primary diagnosis, but it's a secondary issue, right?
It is, yeah, and it's probably, and more people, and a lot of the mitochondrial docs are realizing that treating cerebral folate deficiency does incredible things for improving cognition in a lot of kiddos. You know, it's something I think we missed for a long time.
So I decided to try your spectrum needs about 2 months ago, just for kicks, and tastes great. But my question would be, I started having trouble sleeping, and I wasn't even doing, I was like doing maybe half the dose I was supposed to do. But I was struggling with, well, do I stay on the leucovorin, which they did keep giving me. Do you do that with those high folate levels in the spectrum needs? Is there, you got to play around?
Yeah, the leucovorin dose is much higher than what's in spectrum needs, yeah.
Well, and that's what, but do you put both of them together?
Yeah, because you're probably taking between 10 and 40 of leucovorin. There's one in spectrum needs.
They had me 325 milligrams a day.
Yeah, so in mitochondrial disease, so when the folate autoantibody, which is, you know, when we think of a lot of cerebral folate, it's the 2 milligrams per kilogram, but it's been documented in people with mito, you may have to go up to 4 milligrams per kilogram of leucovorin, so very high doses.
One thing
for some people.
Spectrum needs is meant to be a platform that you build upon. That you build upon it depending on what genes you have, or what symptoms you have, what your experience is. I made it so that it would be safe and effective in everybody basically, so no matter what medications you're on, what supplements, what disease, what mutations you have, what, you know, everything, what SNPs that you have, that it's safe and that it won't give you side effects. I mean, having said that, it's always, if you have a child with an illness, you should probably talk about with your doctor with any product you use regardless. But having said that, it's a baseline product that's helpful for everybody and it's not going to hurt them. You do need to use things on top of that for almost everything with mito. I mean, for one, they'll all need more CoQ. Many of them will need more leucovorin. Depending on the blood level, they may need more carnitine or vitamin D. And then dependent upon, like, you know, one person gets more alpha-ketoglutarate, another person gets more creatine. So it's a good baseline, so if you have the 33 ingredients, you don't have to take 33 separate things, but then you build on top of that for each individual. And it does take 2 to 3 months at full dose before you really get the full effects.
Have other people struggled with sleep at all?
Oh, you know, it's the thing is that when you energize the brain, you get the good, the bad, and the ugly. And if you, I mean, things like anxiety and OCD and stuff like that can get worse when you energize the brain, regardless of spectrum needs or if you take the independent components, because you're giving more energy to those parts of the brain that are considered to be problematic. One thing that you can do is to take it earlier in the day, like take it for breakfast and lunch, and not take it anywhere near the time you go to bed. That's one intervention you can take, obviously reducing the dose. Another thing that you can do is to do things that are common, like 5-hydroxytryptophan, GABA, and theanine, which is actually a product we're going to come out in a few months or so, because a lot of people are over-energized. It's a kind of a calming product. It's just, you know, it's not sedating, it just kind of takes the edge off. Yeah.
I have CVS. I went through it for maybe 8 years where it was like really serious, and like been in the hospital at least once a month, every month, and that hasn't been since I was maybe 12. It's been nearly 3 years, and so, sorry, I'm trying to figure out how to word this, but so there's always a possibility that like in 5 or 6 years that it could get just as bad again, and I have to go through that again.
Unlikely. We don't have a lot of really long-term information. I mean, the basics is a lot of times there'll be family members that at CVS, and I know what happened, a generation ago when they weren't treated. If you go off a treatment, it often comes back. But if you're not on treatment, if it goes away to migraine, it will stay at migraine. It may go to the migraine may get worse. It may go into dizziness. It may get better. But going back to the vomiting is not extremely likely. But anyway, you shouldn't have to suffer with any of this. This is, in the vast majority of cases, this is a treatable condition.
Right over here, I have a question.
Go.
I have 2 quick questions. Dr. Frye, all the moms know you're in town. Rumor is you won't see any kids that don't have an official diagnosis of autism. True or not true?
Well, under the autism label, yeah. So that's kind of, for the autism clinic, it's for kids that have autism. So, but if you have something else, there's other ways of getting it. Yeah.
Other ways of.
Well, so it depends on what you're going to see for. So if you're coming to see me for autism, then you have to have autism.
Right.
But and that's how the clinic is kind of set up. But if there's other things that you want to see me for, you can see me for that, but it won't be under autism.
Right.
Although we'll probably be in the autism clinic because that's just where the space is.
Okay. Second question is, do you often see an overlap between autoimmune encephalitis, those kinds of things, and the mitochondrial kids that you see?
Yeah, most definitely. Yeah, I mean, it's a very complicated interplay between these different things. The immune system is very much attached to the mitochondria, and we actually have some research papers coming out on that right now in kiddos with autism. But independently, there seems to be some very tight connections in both immunodeficiency and autoimmunity, and where those connections are and how they're connected, it's kind of, I think, kind of hard to say, but they definitely overlap. Yeah.
And it's not uncommon when we do exome sequencing that we figure out the problem in autoimmunity. A lot of times, if the white cells in the immune system require a lot of energy, so that's a big hit on it right there. And if they have another gene that can affect the immune system, it's often a combination like everything else. Sometimes we can figure it out, sometimes we can't. But yeah, a lot of them, the immune system's too high and they have autoimmunity, and a lot of times it's too low and they have immunodeficiency, and sometimes they have both. And it is complicated.
I just had a quick question about the research, Dr. Frye.
Yes.
The overactive mitochondria in some of the kids in autism spectrum disorder. Would something like the spectrum needs be contraindicated in that case?
No. So one of the ways, again, mitochondria itself are just on kind of, we're understanding how to treat it. But one of the things that we think of, in the overactive mitochondria, you can think of your car going, at 120 miles all the time. What would your engine need? It would need more gas, it would need more oil, it would need more coolant. So, the mitochondria uses up all these cofactors. So if it's running twice the speed that it's supposed to be, it needs more of these cofactors. And that's what, some people, and this is why a lot of times I don't measure like blood levels of certain vitamins and stuff like that, because those are for normal people. So, norms are for normal people, and we're working with, a system that's not working normally. So we have to think of it in that sense that if this is your regular dose, if your body's working in a different way, it may need twice that dose of something.
Okay, so that might help retain that reserve.
Right. Exactly.
when it's exposed to stress.
Right, and so that's, and yeah, and you have a good point. The question, of course, people have to say, well, so what, mitochondria is working twice as hard. Maybe that's just how it is, and it may be, maybe it can work all that hard, but what we showed is that when you have a little stress, it falls apart. So how do you, protect the mitochondria so when it does get stressed, it doesn't fall apart?
Okay.
One more quick question about that. You were talking about the long-term stress being like inflammation was one of the examples you gave. What was the short-term, is it a toxin that you talked about?
So different types of toxins. So what we've done is we've looked at kind of prolonged, so you wouldn't even call that chronic, but 96-hour exposure we usually do. But we've actually shown in some substances 24 or 48-hour exposures, they could change the way the mitochondria functions, after the exposure is gone. So it depends, and we don't know a lot about many types of toxins and how they actually have their physiological effect. But
what was that one that the study was based on?
Which one? I had a couple of studies I showed. One, we just increased oxidative stress. So we made the kind of petri dish have lots of oxygen radicals, which just causes physiological stress in the cell. Just to have a general stress. We also use chlorphos. Yeah, an insecticide, I believe. So that's another type of toxin that's been associated with autism. Just as kind of a proof of principle that maybe some of these toxins that we see associated with autism, may be working through this mechanism.
So just one other quick question.
Yeah.
So I noticed also one of the studies was talking about during in utero development,
Sure.
being exposed to certain toxins, maternal exposure, would something like that during pregnancy help to protect against some?
That's where we want to find out. Yeah. So that's the the problem that we don't know is that one of the complicated ways of studying pregnancy, right, is because we talk about the kiddo's genes, but what we find is a lot of studies that show that it's the factor in the mother and the mother's genes that are and how they're protecting the baby.
Exposure.
Yeah.
Everyone's exposed.
So you have everybody's exposed, but only some manifest disease, and that's where certain types of variations, probably in genetics, come in, where your body's more or less able to sustain these and protect the. So yeah, so we actually just submitted an NIH grant to answer that question. Yeah, to look at that.
So a lot of the problem on that is if there's a toxin that affects everybody, it would be pulled off the market immediately. It never would have gotten there. But if it only affects 1% of the population, then it would have gone for all the studies and no one would find anything. But 1% is a lot of people. And there's a lot of toxins out there. So altogether, all of these toxins, each of you are susceptible to some toxins and not to most of them. And sometimes genetic analysis can identify that, and usually it can't because it's just no one's really doing that sort of research. But it's just a new area now that we have the capability of looking at the genes in a cheap way, relatively cheap, that we can start to get those studies. But
Just on a side note, I so appreciate your information. I haven't heard so much of what you guys have presented, and I am very excited about passing it along. So I really hope that the slides, the information on your slides will be available to everybody.
And this full presentation will be on our page.
Very, very excited about it. Thank you.
Okay, let's see, how can I ask? My daughter's 17, we're still looking for the needle in the haystack with her. She's had several microarrays done. Nothing came up. She now did a swab, and I think it's GeneDx. Is that right?
is that what they did, like they're doing exome?
I'm not even sure.
Okay.
It's new, because before it was blood work. So we're in the middle of that. The testing should be done like April 1st, somewhere around there. Will that tell me if there's mitochondrial stuff going on?
Well, I mean, the typical way that GeneDx is one of the largest labs in the world, and it is a conservative lab. And if they find something that they believe causes disease, they will tell you about it. but they miss a lot of things because they're looking at it from a traditional medical background, not a functional medical background. They're not looking at it from like a mitochondrial background, and they're not looking at it from a polygenic, what could be risk factors. So if they find something, this is a slam dunk, this causes disease, they should find that usually, not always, but usually. But they only have so much information on your daughter. I mean, I was a laboratory director before. I was sitting in, on my screen. I don't know anything about the patient. You look at the DNA. You can only do so well. You really need to connect that to your daughter and to your daughter's health, and to be there and ask questions and everything. And also, there's like the things that we're talking about are not looked at by traditional labs. None of the things of over mitochondria or anything are looked at. So what we can do, you don't have to redo the sequencing, is I could take a look at the GeneDx sequence and have it reprocessed and look at that.
Okay, that's what I was wondering.
Yeah, that would be, so.
Okay. And we're working, by the way, with Dr. Kruger too, so I think we work with him.
Yeah. So he's great. So he may find something. I think he's aware of the limitations.
And Dr. Alex.
And Dr. So those are great people.
Okay.
But
I didn't know if they would be able to pick out mitochondria.
Well, yeah, I mean, as Dr. Boles says, GeneDx, if you just look at the report, they are going to miss a lot of things because they only call things that are absolutely clear. And that was the nice thing about Portagen when it was around, is that you got guys actually imported stuff and say, this looks a little bit weird. We don't know, So that was kind of nice because you had some information, then you can interpret it. But GeneDx doesn't do that. And so that's when you need medical professionals like, Dr. Boles, Dr. Kruger, Dr. Alex, that are going to go in and actually look deeper into the results.
Okay.
When you say you looked at toxicity, has there been any work on trauma and its relation?
Yeah, trauma is a really important factor, and are you talking about prenatal trauma or what type of trauma? Yeah. All of those things, we're starting to realize, at University of Arizona, we have a whole traumatic brain injury type of program, and that's some of the things that we're trying to look at is what goes on with trauma, because we believe there's a lot more than just the trauma, but all these inflammatory cascades that happen that have long-term effects. That it's not just the trauma that you see there, but a lot of things that happen to the body because of that. So that's something that's actively being researched,
Thank you so much for sharing.
So that's it. So thank you both so much for your time.
We're going to give you a little bit of a break. We're going to go live.
Okay. We're going to go live.
We're going to go live. So we've given people who haven't had the pleasure of being here the chance to chime in on Facebook with their questions, and we're going to go live with Dr. Boles and Dr. Frye to raise questions from people all over the country. So that'll be on our Facebook page as well if you want to see that, so more information. So enjoy the food, enjoy each other. We're so thankful to be here with you guys today and for trusting us to go along this journey with you. So thank you. Jamie's going to ask one favor before you leave.
Yes, if anybody wants to come up and take a photo of This Is Mito. We're doing a project this year with, sorry, let me go back. We're filming people with their This Is Mito stories. So we're going to be just going around asking anybody who wants to film your story, telling your Mito story from your words directly, telling it how it is from your words, not someone else's. We're sharing the stories just so we can capture the MitoAction stories directly from the families that are living them every day. So if you want to turn in your story, you can email me or Kyra. You can get our cards from us. And then if you want to take a picture, we'll share your picture with the stories, or you can actually send in your story too. It's up to you. You can come up to me and let me know if you want to capture it right now or if you want to send it in. We can also send you this to take it another day. I know I just cut you off guard. I've been sitting here for a few hours too. And if you guys want to join the Mom Get Kids, these Mito champions, or hear more about next advice, you can email me to jamie@mitoaction.org. Okay. Thank you.
$70.
$70 and
33 servings, 7 months.
Yeah, I think the number of servings